Hello, my name is Carolina Lean, and my applied project is over mathematical modeling of non-biodegradable polymer microparticles. My chair director is Dr. Vernon, and my two members are Dr. Leung and Dr. Greger. So just to begin with a little bit of an introduction, there have been numerous advancements in the creation of new drugs, but the delivery method for these medications is somewhat lacking. Conventional methods can impact the solidity, the efficacy, and the potency of the drugs that are being delivered. For both intravenous delivery and oral delivery, there can be a drastic increase in drug concentration all at once, and then a very small window of therapeutic intensity when you're doing a drug delivery. So this can introduce a toxic level of medication to a patient in order to receive a therapeutic effect that's very limited. However, microparticles can be utilized to improve the efficiency of safety through targeted release, rate control, and that exterior protective shell. Polymer microspheres are spherical microparticles that range from 1 to 1,000 micrometers in size and can be made from synthetic or natural polymers. These can be used as drug delivery systems that give various advantages to pharmacotherapies, such as the ability to control release rates and mechanisms, have targeted release, and also are able to provide a protective casing to the drug. There are many applications with both long and short-term medical treatments. One example is within vaccinations, which have the benefit of targeting antigen-presenting cells as well as providing a controlled release for multiple doses over time. This eliminates the need for repeated injections in order to have full immunity. The image below shows a representation of a pulsatile release of the polio vaccine. This is able to decrease the need for multiple visits, which improves the health of patients that are in areas with limited access to medical care. Another example is within cancer treatments. Microspheres can be utilized for pinpointing cancerous tissue using site-specific targeting. This limits the detrimental effects that commonly delivery systems can cause to normal cells and improves these effects towards abnormal cells. Release from microspheres is done through leaching or from the breakdown of the polymer's matrix. This is done through diffusion, erosion, or swelling-controlled systems. Different chemical and physical components of the polymer system will alter the mechanism of material release, such as polymer type, drug composition, encapsulation size, and other factors. While microspheres have many advantages and applications, there are certain drawbacks. One limitation is the cost of materials and time commitment, which can be much larger than common drug delivery methods. It can also be really difficult to get approval from the Food and Drug Administration because they have strict regulations on the stability during encapsulation, having a good drug yield and encapsulation efficiency. They also require there to be no adherence of aggregation of the microcapsule. They also need to provide reproducible results and a process that can be scaled to the industrial level so that it can be developed for large-scale use. During development, it can also be difficult to attain reproducibility, and this can cause issues as you move into market. Also, microspheres that are biodegradable polymers can have additional problems with potential dose dumping, some complexities within the drug loading and its efficiency, and being able to have precise control over the release. So with these limitations, it's really advantageous to have a preliminary model before developing a microsphere in the lab and using those materials. So that comes to my project, where a mathematical model can predict the drug release profile for a polymer microparticle delivery system and the effect of modifying different parameters. This allows the delivery device to be optimized before experimentally testing through varying the mechanism, drug loading, and geometry design. This is able to minimize your necessary trials and speed up development, especially when you have limited resources. So my applied project focuses on modeling a non-biodegradable bimodal microsphere system that has a consistent drug concentration throughout. This is done by utilizing fixed second law of diffusion and a modified biwobble probability distribution function that you can see here. The mean, standard deviation, and F1 factor component, which modifies the bimodal, are varied through a full factorial design of experiments. Outputs such as 60% drug release, the total fraction drug release, the diffusional exponent n, and constant k are analyzed for their relationship to predictor values. Future in-lab testing is expected to verify the efficacy of this model. So this is just an introductory video, and I thank you for watching. And if you're interested in a deeper discussion of the results and the mathematical model, please visit my symposium presentation. Here are my references, and thank you again for watching.